Welcome everyone to Module 2 of the Genesis program. With Argentina, we are very happy to have you here, especially our speaker, Sara Holland. Thank you so much for being with us. My name is Luz. I am a volunteer in the area of communications. I am communications lead, and it's a pleasure to welcome you to this amazing program for aspiring biotech entrepreneurs. This is today's agenda. We'll first introduce Nucleate and then give some information about the lecture. Then we'll go on with the lecture and then close for about 10 minutes. And then in some extra time, we are going to have a conversation with our Genesis cohort. So first, here. Tengo un problema. Well, Nucleate is a nonprofit organization dedicated to empowering the next generation of biotech leaders by educating current academic trainees. It is a free organization. All our events and courses are open to all, and it is entirely focused on training and supporting students. Here in Argentina, we are a very large group of volunteers from all across the country. It is now, I think, 30 of us, or I'm having trouble with the presentation. Okay, never mind. We are a lot of volunteers from all around the country, from different provinces, Córdoba, Mendoza, Santa Fe, Entre Ríos, Chaco, and more. And we are led by Marco Mancini and Paloma Ramirez. We are very happy volunteering here, and we feel like we are contributing a lot to the biotech ecosystem in Argentina. This is our roadmap for Genesis 2026. We have just completed module number 1, Principles of Ideation and Diligence. Today we are kicking off module number 2, Intellectual Property, and with this introductory lecture. Over the next weeks, the Genesis teams will also participate in workshops on patent searching and licensing strategies. This year, we had over 130 applicants from all over the country. This made it very hard to make the choice, but these are the teams that we have selected for Genesis Program 2026. First, AgTech team, we have Camilo Alfione, Lucia González Altieri, Lucia Lucas, and Lautaro Otero Mafioni, and they are working on the prompt low-cost biotechnology for preventive animal health, from reactive treatment to resilience. Team Health Tech is Florencia González Morán, Sofía Ujías, Martina Torres Gaufin, and Mariana Díaz Luna, and they are working on the prompt epigenetic reprogramming and cellular rejuvenation, reversing aging at a molecular level. And finally, Food Tech team are Constanza Agostina Guimaraes, Vanessa Beretta, Paula Otero Signorelli, and Francisco Marco Berberian. And they're working on synthetic biology as a driver of innovation in food production, design of new ingredients, proteins, and systems. And we also have sponsors for this year's program. First, we have Crossing Over LATAM BioExpo. In this event, the students will be presenting their final projects. And also CPPR, who will be working with us, providing their services with a value up to $1,000. So I would like to formally introduce Sara, our first speaker for Module 2. thank you again for being here. So she is our intellectual property expert. She's advisor to the biotech and synbio startup community, and she works at Potter Clarkson. So whenever you're ready, please go ahead. I'll stop sharing my screen so you can share yours.
I'm gonna go. Let's see. This one. Can you see that? Has that shared?
Yes, fantastic.
Okay, cool. Thank you. Well, thank you very much, everybody, for having me, and thank you for turning up to come and listen about IP. Sometimes, you know, it's not the most interesting of things, but it is really important. And I think it wasn't until I left academia that I realized actually how important it is. I know what this is doing. Okay, so just to introduce myself, my background is I was an academic for quite a while. So I did my degree, PhD, and 8 years postdoc at the University of Nottingham in England. All my research was basically yeast molecular biology. So I was looking at how centromeres work, and then looking at how mostly chromium is toxic to yeast, doing lots of fundamental molecular biology. And then I got to a point of realizing, although I really enjoyed the science, I all of a sudden just couldn't stand doing the lab work anymore. I could not do more fairly routine lab work that probably wasn't going to work, that I was going to have to repeat. But what I really did enjoy doing was like learning about the science, coming up with ideas, being quite creative in science. So I looked for something else, and I left and became a patent attorney. So I've been at Potter Clarkson nearly 14 years. I'm a partner now. So my job is to help some academics I work with and some very early stage, mostly scientist founded companies. So people like you who've gone off and set up a company, I help them come up with their IP strategy and protect their inventions with patents. So my core role is to work with people like that to draft patent applications, get them filed, argue against the examiner as to why you should get a patent. They will say you shouldn't get one. We will say, yes, you should. And I'm going to go into a little bit about how that all works in a minute. To hopefully get you something granted that covers what you're wanting to do commercially, and also kind of creates this space to stop other people working in your area. And when I worked in research, I always thought the way I would have an impact on the world is by publishing my research in a journal. Some big tech company would read it, they'd think, wow, this is amazing, and then they'd go off and develop a new drug or whatever based on my research. It was as soon as I left the lab, I realized unless you've thought about intellectual property, these big companies or nobody really is going to care, because it's only when you have secured these intellectual property rights that gives you the kind of space to stop other people working in your area. So if you want an investor to give you $1 million to develop your new drug, why are they going to give it you if once you've developed it, somebody can just copy? So a lot of the intellectual property work we do is kind of protecting your space so that you can raise the money. And ultimately, somebody somewhere has to make money, whether it's you, whether it's a drug manufacturer, whether it's a ag tech company. And you know, that is just how it works. So I will talk a little bit about, I don't know if I need to click that or this. There we go. Different types of intellectual property. I'm going to focus on patents because that's the main one that's going to be relevant to us on this call. So intellectual property, lots of different types, and they serve different purposes. So there are in most places there are something called design rights. So designs protect just how something looks. So it's nothing to do with how it works. It's just the physical appearance. So the common example that people use here is a Coca-Cola bottle. So the shape of the Coca-Cola bottle, or perfume bottles, they'll be protected by design rights. So it's gives you, whoever owns the design right has the right to stop other people making a bottle that shape. Which, if you're big and like Coca-Cola, you don't want people nicking your shape and pretending to be Coca-Cola. Similarly, for the same reasons, we've got brand protection. mostly through trademarks. So trademarks are used in loads of countries, mostly to protect the name of a company, but it can be like names of products. So you might have McDonald's, and then under that you've got your Big Mac, and you've got your different product ranges, and lots of companies will do this. Drug, therapeutic drugs, they've got trademarks around them. And trademarks are not just the name, the word, it can be the logo, it can be color schemes. So a certain petrol company has a color scheme that's a certain green and a certain other color, and they protected that color scheme. So nobody can use that color scheme in the field of petrol. Confidential information, people do overlook this as a form of intellectual property. It's just something and that nobody else knows, but it is quite often very valuable. And a lot of the companies we work with, they will have patents, but they will protect some of sometimes the most valuable stuff using confidential and trade secret protection. So this only works if you can keep it confidential. And I think I've got a slide in a minute, but say we've engineered a new bacterial strain for some ag tech product. The strain's going to go out there. You could sequence it. You can find out what, mutations and things I've made to it. So that's something that we can't keep confidential, because it's going to go out there. Somebody can reverse engineer it. So that's not suitable for trade secret protection. You'd want to protect that with a patent. Copyright, probably all know about. That's kind of how, music, art, literature, things like that. Data is another form of intellectual property that people overlook. It's very valuable, especially these days. If you've created a big data set, that's yours. You can either keep it totally confidential, or you can let other people have access to it for a fee. So that's another way to kind of make a revenue. But the one I'm going to talk about mostly is patents. So patents is the science bit, really. Patents protect the technical and clever way something works. So nothing to do with how it looks. That's just your designs, but patents are the technical, clever bit, which is, for me as a scientist, much more interesting. So this slide just shows how even in one I say simple, I don't know how a battery works, I'm sure it's not simple, but in one little product, there's all these different intellectual property rights coming together. So you've got designs with the appearance and maybe the color scheme, so you've got the gold and the black. Copyright around the text that is in the packaging with the battery. There might be some trade secrets on how you actually make the thing. Trademarks, obviously the name, and maybe some other things associated with the actual branding. And then the patents are the clever bit, how it actually works inside and all the bits and bobs inside. So for probably most of my clients in the biotech space, we're not really looking at designs, because we're not really interested in how things look. Designs are useful for medical devices, because you can protect the shape of, like a catheter or something like that. But most of my clients will definitely have patents. They will have trademarks. They will have trade secrets. They probably will have some copyright, but aren't really too fussed about it. So I started talking earlier about how I hadn't really realized the importance of intellectual property while I was doing scientific research. So why should we bother? Because it is expensive. So one thing when I'm talking to academics is, I stayed in academia too long, and I probably should have left earlier. I'm glad I didn't leave earlier, because I wouldn't have got this job at the right time. So it all worked out. But, I probably should have left earlier, because particularly these days, I'm sure it's the same in most countries around the world, an academic career is not for life anymore. In the UK, it used to be if you got to be a lecturer or got your own research lab, that was it. But certainly, even in the university where I was, not University of Nottingham, they are at the minute probably going to make quite a lot of people redundant. And this is including professors and a lot of the research staff. So, academic jobs are not jobs for life anymore. So one thing you could do, if you're really interested in the research you've been doing, is form your own company and spin out. So you could carry on doing the research and pursue it, but just in a commercial setting instead of university. The other key thing, especially for academics, to think about why you do want to think about IP is that translation piece. So like I said, I thought if I publish it, that's my contribution to the world. But actually what the world really wants is things we can hold and use and use to make the world better, treat people, or just have fun with. And the only way you get that from your bench out into the real world is through translation and commercialization. That's the main reason. So if you do want to have an impact on the world, don't think you've done it just by publishing a paper. You need to think about actually getting it out there. And then there are other reasons. So if you are, like I said, if you're going to set up a company, investors will want to see if you have got your technology protected, or if it is protectable, so that they can, like I said, if they give you the money, you're going to have this monopoly space in which only you can work it. So then the investor will get a return on their investment longer term. It is especially a biotech startup's most valuable asset. So if you go set up a company tomorrow, you probably don't have your own lab space. You're probably having to rent or beg bits of lab space. You don't have your own kit. There's lots of things you don't have, but what you do have is your idea and your intellectual property. So that is the thing that you will be raising money based on. It's the thing that you would be acquired for. So one of our clients, they're in the antibody therapeutic space. It is quite, a space where things are very expensive. But they were acquired a few years ago for, I think it was like $2 billion. And they don't even have a product on the market. All they have is a bunch of patents that protect their lead drug. And that's what they were bought for, the rights to that drug. And then, you do need to think about it early, and if you don't think about it properly early enough, you can make mistakes which you can't come back from. So it's definitely worth learning a little bit, knowing what you can and can't do, and thinking about it properly. So I've talked a bit about patents, but not really said what they are. So patent, if you get it granted, and it's not guaranteed, but if you get it granted, you will get a 20-year right in which you can stop other people working your invention. So we often refer to this as a negative right, because it doesn't mean if I've got a patent granted to my engineered yeast strain, doesn't mean I automatically have the right to make and use it myself, but it does mean I have the right to stop other people using it, which can be a confusing thing to get your head around. But that's because if I make my engineered strain and I'm putting 2 mutations into the strain, great, I've maybe created something new and clever, and I can protect that myself. But somebody else might have got a patent to this mutation, and somebody else might have got a patent to this mutation, which means that if I use them and make my strain, I'm infringing their patents. So that's something companies need to think about. And it's when we talk about it in my job, it's called freedom to operate. So although I might have got my new funky strain myself, am I free to make it without infringing somebody else's patent? So I do have a slide later on this, I think, but it can be difficult to get your head around. But basically, even if you're not patenting anything yourself at all ever, you still need to think about it, because anything you do could infringe somebody else's patent. So there's 20-year right, so it doesn't last forever, which is part of the reason you get this monopoly, because if you had a monopoly for forever, that probably wouldn't be fair. But you get 20 years in which to try and make your money back. And during that time, most companies will be trying to iterate and make new improvements to file new patents to make their technology better. Then after the 20 years, anyone can use your invention, and it's free. This is what generic drugs are. So once the patented drug has run out, then generic companies can enter the market and sell the drugs cheaper. Patents are territorial. That's one thing I didn't know when I started this job, coming straight from academia. So if I want to be able to have the right to stop people using my invention in America, in the US, I have to have a US patent. Same in Argentina or wherever you need a patent in that country. So you have to, there's various timelines, and there's a certain point at which you have to make that final decision. And once you've gone past that, you can't go back and add countries. So there's a point at where it's now or never. You can drop them later, but you can't add them back. So the countries people want to think about are basically where's your customers? Where are you selling it? Where are you going to make it? Where do other people have the ability to make it and, generate copies that you don't want people to do? So there's lots of places, lots of things to think about when you try and think of where to file. There isn't a worldwide patent. Nobody files everywhere, because that would be a lot lot of money. Not even the big, the big pharma companies with lots of money file everywhere. They're quite strategic in where they file. So that's something we have to work on with our clients, trying to decide what's a reasonable list of countries to file in without spending all their budget, because startups have a lot more things to do with their budget than spend it on patents. So all of this feeds into your IP strategy. And then in patents, there are lots of different types of things you can protect with a patent, and some are surprising sometimes, I think, to people. So all the different categories of thing we can protect, so products and devices. We can protect methods. So methods can be manufacturing processes, cell culture methods, methods of using something, all sorts of stuff. And then in the product side, we can protect proteins, we can protect nucleic acids, we can protect microbes. And in most countries, these can be engineered, so not naturally occurring, which most people think is fine. But we can also, for example, in Europe and lots of other places, protect naturally occurring proteins, nucleic acids. We can't protect them just because you've found them. So if I find a new yeast species on my laptop, I cannot get a patent to it just because I found it and it's new. But if I can show that new yeast species, I don't know, makes a black dye, or makes something that will be useful, or it's a better host chassis for expressing therapeutic proteins, then I can protect it. And I can protect it just as the yeast. You don't have to protect it for those particular uses. But as long as we can show it's got a technical use, we can protect it. Software we can protect in some places. Software is a funny one, and I am definitely not an expert on software, but it has to have a technical use. You can't just protect a computer program because it's a computer program. It has to have some sort of technical output. So yeah, lots of different things you can protect. It can take a while to grant. This says 2 to 4 years. I think it's mostly longer. And most of my clients and those in the kind of therapeutic space, they're happy for it to take a long time, because the only advantage in getting something granted quickly is if you want to sue someone. So if I think you're infringing my patent, I can't tell you to stop until my patent's granted. That's when I might want to speed it up. But most of the time I'm happy, because it does cost money. I'm happy for it to plod along and get granted when it gets granted. So to get a patent to your invention has to be new and it has to be inventive. And if you don't take away anything else from this, remember your invention has to be new. So if you talk about your invention anywhere, it is no longer new. And I've got a slide in a minute. So keep details of your invention confidential until you've filed an application. Yeah, so this is an example of prior art. So when I say your invention has to be new, so say I file an application today, my application and my invention will be judged against anything that has been made public before today. So yesterday, any time before then, anywhere in the world, in any format and in any language. So in an academic setting, this can be poster presentations and abstract books, conference talks, that kind of thing. It can be theses that have not been embargoed. It can be website disclosures. Journal articles is obviously a key one, because that's why we're all in academia, to publish papers. So it can be anything. And this is just an example of a UK patent where a comic in the UK, the Beano, was cited against an invention that was a doggy doorbell. So the Beano had described a doggy doorbell years ago. Somebody tried to protect a doggy doorbell with a patent, and they couldn't have it because this comic makes it not new. In academia, it's really important to know your own disclosures do create prior art. So most of the time, like I said, academics, we're talking at conferences, posters, journal articles, that is all creating prior art. And most of the time when I work with universities and I've got an academic with an invention, the main problem we have is against their own papers that they've already published. So if you are publishing, just keep track of what you're publishing. And remember, if you do want to protect something and you do talk to a patent attorney, make sure they know what you've already disclosed, because it's better to know, because we can draft around it, than suddenly find 2 or 3 years later that an examiner's found your Nature publication that you forgot to tell us. So I said your invention has got to be new. So it's got to be different to everything that's been published before you file your application. But to be new, it only has to be, you only need one difference. As long as it's different, it is new. So it could be, let's see if I've got any examples. No. So in a minute, I've got some slides about newness. But for my invention to be new, it could be one amino acid different to something else. It could be a different temperature or pH. So it could be something that's quite small. And certainly when I was doing research, I had the odd idea about stuff that I was working on. I know I thought, oh, somebody's done this before. But now I know for somebody to have done that before, they have to have done exactly the same thing. So if yours is different in any way, and particularly if that difference makes it better, then you've got something new and potentially inventive. So the invention's got to be new. It's also got to be inventive. Otherwise, it's not an invention. It's just some new thing. So inventive step. Novelty and whether your invention is new is quite standard across the world. It's either the same or it's different. It's quite black and white. There's no wiggle room. The way inventive step is judged does differ quite significantly in different countries. So, for example, in Europe, we look at a lot at the European Patent Office, and the examiners will look at a lot of whether there's motivation. So filed my application, we've got all this prior art that's been published before we filed our patent application. Is there anything in there that would motivate someone we call the skilled person to arrive at your invention? So in Europe, they quite often look at, the examiner would normally find two documents. One is quite close or the closest to your invention. Is there anything in there that says something like, oh, it would be a good idea if we tried these other mutations, or it might work better if we tried it at a higher temperature, and then you try it at a higher temperature and it works. In those cases, your invention is probably not inventive because you're just doing what these early documents are telling you to do. But quite often that isn't there. And the European test is quite useful from a patentee point of view because, you know, you've got two documents that if you did put them together, you would get your invention. But because there's nothing telling you to put them together, you're okay. And your invention is classed as inventive. The US in particular is quite different. The US examiners are allowed to piece together lots of different documents. So sometimes 5 or more documents. And they don't really rely on motivation. So if something exists, they say it would be obvious to put it together. So DNA exists, tubes exist, this exists, this exists. It would be obvious to put them all together. And you wouldn't get that in Europe. So the US can be quite challenging sometimes to get something granted. So like I said, we look at this, the inventive step is judged through this kind of made up skilled person. So you're all thinking up here, much higher level of thinking than the skilled person. So you're all kind of creative, doing research, thinking about science, and you're coming up with new things. It's your kind of job or your role. The skilled person is much more down here, and they can follow protocols. They can basically do what they're told, but they can't make that kind of creative leap. They can't think on their own, oh, what if I tried this? If a document said, go and try this, they will go and try it. And if that makes your invention, then you're not inventive. But without that, again, motivation, this skilled robotic person wouldn't do that. So two things. If you think you might have got something that you want to think about patenting, firstly, is it different? And remember, one difference is enough. Secondly, don't dismiss it because once you've come up, all of us, once you've had a thought, then it becomes obvious. We use this hindsight knowledge. So if you have come up with something, try and think, well, actually, it might be obvious to me because I came up with it and now I know about it. If I put myself in the mind of this skilled person and I look at what is already known, is it actually obvious? So don't dismiss something you might want to pursue in a patent just because to you it's too easy. Talk to somebody who can help you think more like this skilled person. So these slides just have some examples of what might be novel and inventive, and I'll just whiz through them. So you might have a different, again, so you might be expressing a protein, you might just change the host species, and you might think nothing of that. But that is new because it's a different host species. And if that change makes it, I don't know, I've got reduced immunogenicity here, but it could be improved expression, anything like that, then that could be inventive. Similarly, you've got the same host species, but you've knocked out a gene. What has that done? It's new. If it does something useful, it could be patentable. This one is like one of the smallest changes. So one of our clients does lots of PCR diagnostics and makes probes for RT-PCR and things. And I think the prior art, all the probes have the fluorophore right at the end. They found if you move it in a few nucleotides, you get much brighter fluorescence and a better reaction. So again, that is literally a tiny change, but it was enough to get something granted, because you couldn't predict that moving it would have such an effect. Similarly, just different growth media. A 1-point mutation in an enzyme can affect the activity. Combinations of known things. So a product itself to get kind of a product claim, you don't have to have a brand new product. You can have combinations of known things. So as long as it wasn't obvious to put them together, that can be patentable. Again, different buffers. So these are all quite small changes, but could be enough to get patented. These are just some chemistry examples that I kept in, but I can send you the slides. Again, different, quite small things, different temperatures, different combinations of monomers, things like that. These are these are very clicky slides. They were like engineering examples, and then software. Like I said, it's not my area, but I did include these in case anyone wants the slides. And if you do want to talk about software, I can find a software person who knows about patenting software. It's not me. So in terms of some other practical points, if you want to file in a biotech or life sciences therapeutic type space, we do need a bit of data normally. It depends what the invention is, and I've been looking into this quite a lot recently. So if you say we've got let's go with a PCR based method. You've come up with a new PCR diagnostic, maybe a certain arrangement of the primers or certain temperatures, all that kind of stuff. And once you've come up with it, it kind of makes technical sense. We know how primers work. We know how oligonucleotides work. As soon as you've come up with that arrangement, on paper, it should make sense. In those kind of situations, you don't necessarily need lots of data or even any data to file, because it makes sense, Similarly, in the engineering space, they quite often don't ever make or haven't made whatever it is they're wanting to protect by the time they file an application. They can just draw it. They can draw a new stapler, and because the laws of physics are known, if you put it in a certain arrangement, then it's going to work. Similar to some biotech and life science inventions, because the component parts are so well known. But in other areas, such as if you have a therapeutic invention, you normally need a bit of data, because it's not necessarily predictable that this new compound will work to cure cancer, or it certainly doesn't need, if you are in a therapeutic space, it does not need to be animal model data. Even just some in vitro data showing this drug binds to this receptor can be enough, because then you've got the rest of the story that, well, this receptor is involved in this, blah, 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 blah. So there's some data is quite useful as well. So as well as being novel and inventive, we also have to make the actual text of the application sufficiently disclosed. So this is another legal requirement. So the whole point of the patent monopoly is that I get a 20-year monopoly for my invention in return for telling the world how my invention works. So patent applications get published after 18 months. So anything I put in there becomes in the public domain. So I'm kind of sharing it with the world. And I have to do that in a way that anyone could work my invention. So I can't patent something if I don't tell people how to do it. So I have to, again, put in lots of information. Might need to include protein and nucleic acid sequences, particularly if you're engineering, or rather, particularly if it's a wild type microbial strain or a mammalian cell line, things like that. You might have to deposit them at a national depository, because at the minute, so if I evolve a yeast strain to something better and I want to protect that, I can't really describe that in words at the minute. We could sequence the whole genome, we could put that in an application, but that doesn't really allow someone to recreate the yeast, because we can't create a yeast yet from just a nucleic acid sequence. So to fulfill the requirement for microbial things and cells, we normally have to deposit them under what we call the Budapest Treaty. So it's a specific patent system, which means I can deposit my strain, nobody can access it, and it's still confidential, but I get the information I need to then put into a patent application. So it's just something to be aware of, because you do have to do that before you file an application. And if you don't do it, you might have got the most amazing strain in the world, but you won't get a patent granted because you haven't shared it with the world technically. So this is going back to what I said a minute ago, really, about plausibility. So again, if say therapeutic, if I don't have to show animal model data, but I do need to make it plausible. So if I can show some mechanistic data that makes it make sense that it would work, that can be enough. And again, data is important for a lot of my clients are, again, engineering microbes, and they're knocking out 3 or 4 genes, and we're filing quite early before we've even got the data, and it's not the best situation to be in, really, because it might be that, you know, those genes don't actually do anything when you knock them out. So it's a difficult one, knowing when to file. So some clients want to file early because they want to be able to talk to investors and share what genes they're knocking out. And if you do that before you filed your application, you risk your patent application then not being new because you've made some stuff public. So it's a hard one. But the data you need in a patent application is nothing at all like you need in academia, which was one of the other things that surprised me when I started this job. You're used to repeating things, getting all your gels perfect, so you know they can go in a journal. No one cares in patents. This is a gel from my thesis a long time ago. You won't put that in a paper, but for a patent application, it shows what it needs to show. That's all we care about. So if you're doing experimental work for a patent application, make sure it's controlled properly, make sure it shows what you need it to show, but don't waste your time and your company's money repeating it to make it perfect, because nobody cares. Oh yeah, the kind of data as well that's useful for us is quite boring for you and the scientists. I suppose if you think of a drug and I show, I want to claim it for treating cancer, and I show it works on a breast cancer cell line, great. Patent examiners will often only let you have a granted patent that's quite close to what you've shown experimentally. So this came up a lot in some of the stuff I worked on a while ago. If I've shown this drug works on breast cancer, fine, I can probably have that. But the examiners would say, well, all cancers are different. They're all slightly different. You can have it for breast cancer, but you can't have it for cancer more generally. Whereas if I had tested it on a few more cell lines, that gives us a reason. So it works on 5 different cell lines. There's no reason it shouldn't work on everything. So it gives us ammunition to argue against an examiner as to why we should have the broader claim scope rather than just what you've shown. And this applies to pretty much all biotech inventions. The more, and I kind of think of it as sideways broadening data. You know, you've shown your drug works on breast cancer. Maybe all you care about is breast cancer. You're not interested in anything else. But from our point of view, that's quite narrow, or it's narrower than you could have got away with. So if you are thinking about filing a patent application, just take a minute to think about what kind of data will be useful to support that, because a lot of it you could do at the same time as doing your other experiments. Instead of using 1 cell line, use 4, just chuck some more stuff in. So how much data you've got is tied very much to how broad we can file and claim your invention. So the more data, the broader the claim scope. The other thing to mention is, I won't get into it too much, but we have what we call the priority 12-month period. So if I file an application today, 4th of August, anything before the 4th of August 2026 counts against me for prior art purposes, and the examiner can say my invention is not new or inventive because of all this stuff. If we didn't have this priority window, I would today have to decide all the countries I want to file in, So they're all filed today. So I'd have to think, oh yeah, I want to file in Argentina and the US and China and Japan, and that would cost me a lot of money. I don't even know if my invention works properly yet because I've not had time to test it. I've not got investment. So that's a bit rubbish. So the way the patent system's set up is you get this 12-month kind of free priority year, really. So if I file something today, I can file another application in 12 months, so 4th of August 2027, and claim the date of today's filing. So even though I file it in 2027, for prior art purposes, it's as if it was filed today. So nothing published in this next year will be cited against me. And that gives me this extra 1 year period to speak to investors, work out how well my invention works, how broad do I want to go? Do I want to file it everywhere and spend lots of money, or do I only want to focus in certain key territories? And then on top of that, you might have heard of a PCT, International Filing System, which I won't go into in any detail, but that gives you another 18 months on top of that. So basically, if I file something today, I have 30 months from today until I have to make the final decision using this PCT system and the priority year. So it's quite a long time to, you know, check if it works, speak to investors, and decide what I actually want to do. So that was mostly everything I was going to talk about on patents. The other side of patents is trade secrets. So if you want to file a patent application, hopefully you have some confidential information, because if it's not confidential, then your invention is not new. So you've got some secret information. It could be in the kind of techie space, it could be certain process parameters that you run your method at. It could be mutation. So if you've got an in-house strain, it could be mutations you made to your strain to make it better. Lots of things. It can also be non-technical stuff. So it could be a recipe. It could be commercial data like price lists and things, anything that's got value to your company. So a lot of what I do with my clients is working out, what do we want to patent, which is going to get public and it's fine. What do we want to keep confidential? Because as soon as people know, you know, they can use that to their advantage. And that could be things like my method runs better at 40 degrees instead of 20. If I publish that in a patent, fine, I might be able to get a patent to it, but it's going to be very difficult for me to know if you're doing your process at 20 or 40 in-house. You know, I'm not going to be able to know, and I'm not going to be able to enforce my patent. So that's the other thing to think about. As well as, is it easy to reverse engineer? Am I going to be able to determine if you are infringing my patent or not? And if not, then I might want to keep it confidential anyway. So trade secrets tend to have a bit more of a special status than just normal confidential information, and there's more legal support and things around trade secrets, and they're more enforceable. But a trade secret has to be secret, has to have value because it's secret, and you have to have taken reasonable steps to keep it secret, and more steps than just normal confidential information. So you might have your standard confidential information, but if you want a trade secret protection, you need to kind of demonstrate, for these, only certain people have access to them, it's under a password, blah, blah, blah. So you have to kind of show this kind of special level of protection, really. But trade secrets are very valuable. And then I've nearly done, so I haven't spoken for 90 minutes, but I'm hoping there's going to be some questions. So the other thing I just want to talk about is inventorship and ownership. And it does differ between countries, but typically the inventor, so an inventor is somebody who comes up with the core concept or who really helps develop that concept. An inventor is not somebody who does all the work necessarily. So if you think of journal authorship, normally your first author will be the person in the lab doing all the work, and the final author will be the PI sat in the lab, hopefully coming up with ideas, but not always. In that situation, the PI will be the inventor because they're having the intellectual ideas, and maybe the PhD student who's just implementing those ideas might not even be an inventor if they are just doing experiments that somebody's told them to do. So the inventor is somebody who's contributed to the intellectual creation of the invention. So it's different to a journal authorship. the inventor is often the first owner, but in many places, including the UK and lots of other countries, the default law of the country is that if you're employed, your invention will automatically belong to your employer. So if I invented something at Potter Clarkson would own my invention. If I invented something as a postdoc at the university will own my invention. And this is something that a lot of people I talk to who are kind of PhD students or postdocs, and they've come up with something and they want to take it and go off and set up their own company. And we have to tell them, well, actually, the thing you've come up with, because you came up with it while you were employed at the university, is not yours to freely take. So that's when you have to talk to your tech transfer office or other commercialization office and try and work something out. And it might be the tech transfer office says, oh yeah, that's a really good idea, let's file a patent on it and then you can help commercialize it. Or they may say, no, we're not interested in it. And hopefully they may then offer it to you if you did want to go and do something with it. But it does differ a lot between different universities. I work with quite a few different universities and they're all different. So if you do have an idea, first check your employment contract. What does it say when you started working at the university? Did you sign away your rights? Is there a default situation where they do employ, they do own it? And then the other thing is, who are you funded by? So sometimes, although you're employed by the university, you might be funded by a different research institute, and they may have terms in the, you know, if they're giving you or the university money, there might be terms in there about who will own the IP. So it can be, I know it's a big headache for TTO officers to sort this out, especially when you've got academics from different universities working on the same project. So if I'm working on a project, then my university will own my part of the invention. But if I'm collaborating with somebody, then there might be other universities and other institutes involved. So it can be quite nightmarish, I think. This is just a quick slide around non-disclosure agreements, because a lot of people rely on them. And I won't go through this in lots of detail, but if you want to talk about your invention in any detail, firstly, try and do it on a need to know basis. Who you're talking to, do they need to know the level of detail I'm about to say? So if I've engineered Saccharomyces and I've knocked out these 3 specific genes, does this person need to know what genes I've knocked out? Or do they just need to know, I've engineered a strain that is really good at making product X? That's probably more the level they need to know. They don't need to know the core clever bits. If you do need to go into that level of detail, do it under a non-disclosure agreement or confidentiality agreement. People have standard ones of these. There are various things to look out for, which is listed here. But the problem with things like NDAs, well, 2, if I tell you the secrets of my invention under an NDA, I can't physically stop you going off and doing it. I can't lock you in a box and say, no, you must not do my invention. So once I've told someone, they can do it, or they can share it with other people. Secondly, if I did think you've stolen my invention, I would have to sue you under breach of contract, under breach of the NDA. So it's not necessarily a perfect solution. So don't rely on them too much, but certainly think about putting one in place if you do want to talk about your invention to anybody. And then I won't really go through this. I tried to explain freedom to operate earlier. This is something that companies always have to look at, and they don't have to look at it straight away, but soon you would want to. So are you going to infringe somebody else's patent? Is the strain I'm wanting to use patented? Is the method I'm using patented? It could be anything really, but you would want to focus on the like the mission critical bits of your technology that you want to do. Some bits you could probably change and use an alternative if there was a problem, but some bits are going to be core, like, oh, it has to be this strain because this is a strain I've always used, this is one we've engineered. If there are patent rights around that strain, then you're a bit stuffed. So that's the kind of thing you'd probably want to find out sooner rather than later. And then I think that was the end. So I can stop sharing. If anyone has any questions or wanted me to go into anything else, it was a quick whistle stop tour, I know. Please let me know.
Sure, thank you. It was great. Any questions? You can open your mic or you can type it.
We can translate if anyone is a bit skeptic about the English.
Yes, sure. Mariana, go.
Hello, Sarah. Thank you so much for your presentation. This has been really helpful. I have a question. In this program, we have to think about a process or something to come up with a prompt. So we are thinking on designing something. I'm on a health team. I want to know how can we find these patents? I've been trying to search specific patents for something I want to use in my solution. And the only thing I know is it's been patented on 2006. And as long as you said before, the patent only, it's only valid for 20 years.
Yeah.
So it's been 20 years now. So I can use it now. I have to search if this is patented again for 20 more years.
Yeah, I guess, yeah, there's a few things. So you don't know the patent number or anything?
I don't know how to search it. I've been trying to search it, but I don't understand the things I found.
If you want to send me some information afterwards, I'll try and have a little look. I can't guarantee because sometimes a patent search thing is not very good. But if you want to send me some stuff, I'll have a look. Yeah. So there's two things. If it was patented, if it was filed in 2006, it should be about to expire. But there are some countries, and it depends what it is. There are some countries, mostly the US, where if there has been a delay in the examination, you get bits added on, and that can sometimes be years. So it can be quite dangerous in the US to think, oh, it's been 20 years, it's gone, because it could still have quite a few years. Also, if it's a therapeutic, there are, in most countries, well, in the UK, it's called a supplementary protection certificate, but for, I can't remember what it's called in the US, like PTE or something, it's an extension companies can get to kind of compensate for the delay in how long it takes to get your product authorized for regulatory approval. So in the UK, you can get an extra 5 years patent term on top of the 20 years. So it just because it's been 20 years doesn't necessarily mean there's no protection out there. And then the other thing to think about is it will still be prior art, obviously. So that patent will have published in like 2027 or 2028, 2007 or 8. And that will be prior art against anything you wanted to file on. So from a freedom to operate point of view, you're probably okay, but we don't know until we can check the relevant patents have expired. But from a patentability point of view, it will still be there. And then, like you said, people can protect improvements. So it might be they filed something in 2006 I don't know what it is, but say it's a compound. And then a few years later, they file something to a specific medical use for it. So companies do find ways to extend the protection by finding new, they might have reformulated it or lots of little things like that. So if you want to email me some details of what you're trying to look for, I can have a quick look, but I can't guarantee I will find it. But I can try.
Thank you. Is there some useful page that you could recommend us to find these patents? I've been using Google Patents, but I'm not sure.
I think Google Patents is as good as the one I use is a Spasnet. Hang on, let's find there. Google Patents, all the patent databases should have everything, really. This is where I tend to search. Can I put this in the chat? I can't I?
In the meanwhile, just a heads up from the Nucleate team, and we'll have a few more workshops about searching patents and the different, searches you can use. So no problem, and we'll continue working on this.
Good. It is difficult sometimes, even if you know the number. The way the number's formatted slightly differently will mean it doesn't come up. Like you might have to drop off the first letter or put a space in, and yeah, it can be a pain. But the other thing if you are, and I'm sure they'll cover this in the other sessions, but particularly if you're looking for a patent filed by an academic, so say I don't know, earlier, a Nature paper, and we know they filed something from it. In the website I sent, you can search inventors. So I would search for the first author, and I would search for the last author, because they are probably going to be named as inventors on it. So that should hopefully pull it up. But also remember, patents only get published after 18 months. So you might find a paper tomorrow that's really good and it's only just published, but you're going to have to wait 18 months to read the patent that goes with it.
Sara, I have a doubt regarding the cost, because in one slide I read that you put that patenting is expensive, but I was wondering how expensive, and also that it depends on each country, I guess.
Yeah. Yeah. So I think for us, we would charge, and this is just getting into, I do what I can to kind of keep costs as low as I can for startups and people I know are kind of just starting out, but it could be between £6,000 and £10,000, like GBP, to draft and file it. And then it has to go through, I can send a document with some costs on, actually, which might be just generally useful, because it's also got a timeline in there I've put together for startups. So that would be the cost of file. Then you've got a few years of examination, which might be several lots of, say, £3,000 or £4,000. So it does add up. That's why it's good sometimes if things do take a long time, and then you can start raising investment or grant funding.
Okay. And I had another related question. Is there some markets or some countries that you must patent your invention in that markets, or no? You just have to choose it.
You can. It's entirely up to you and your company where you patent. You would probably want to protect in your home country and where you're making it and where your customers are. So if I don't know, I can't think of an example, but if the only people who are ever going to buy my product are people in Japan, then if I don't have a patent in Japan, anyone can sell my product to people in Japan. So you want to have a patent there, but you can't have one everywhere. I mean, you could, if you had infinite money, you could, there's nothing to stop you doing that, but nobody would ever do that.
Okay, thanks.
All right. Any more questions? Okay. So at the minute, and this has been tested in a few countries, an inventor has to be a legal person. So it has to be a person, or an inventor has to be a person. I don't know how that will change. I hope it doesn't change. I hope there will always have to be a person involved, even if it's just at the level of, directing the AI tool to do something and then testing it at the end. You normally still need some wet lab to prove it works. Because I think it would be quite sad if all science was just done by computers and there was no like magic anymore. But yeah. So at the minute, and so I don't think it would really matter too much, but I think we would advise saying you're using AI as a tool, not AI invented this. Yeah. Any other questions? I'll send on a couple of documents if you want to share them after, and I'll send these slides as well.
Yes, Mariana.
Sorry, I have one more question.
There you go.
Does this invention have to be proven in only humans, or it can be proven in mice models, and it's also can be patentable?
Yes, yeah. So you mean for a human therapeutic?
Yes.
Yeah, like I said, to go obviously into the clinic and all that kind of stuff, you're going to have to do human trials. That regulatory approval process is very separate to patents. The patent world, we're looking at, is it plausible that it will work? And yeah, ideally you might have some mammalian data. Nobody really has that, because if you leave it too late, you've missed the chance to file, because too many people know. So like I said, it might not even be any animal data. It might be cell lines in a dish. It could be, as long as it kind of establishes a link between your drug or whatever you're doing and the effect. So if activating a certain receptor is known to cause apoptosis and cell death, and so useful for treating cancer, if you find a drug that triggers that receptor, that's probably enough. So your new drug is something you've designed. Nobody knew that before. It's new. It's got a technical effect, and you've shown it works. You've got some data to support it because you've shown it binds to the receptor. The rest is explainable by everything that's gone before. So yeah.
Thank you.
Yeah, the only time I think it's only come up, and it hasn't really come up yet. One of my clients, they are doing IVF type work, and they've done it in mice, and they are doing it now in humans. But I think there was some concern. I suppose it depends on what model and things you're looking at. Some concern that the mouse system doesn't quite replicate the human. So I suppose if you're using it in a model where there's that kind of doubt, or you might have doubts, but as long as those doubts aren't out there in the world, that's okay. So if the examiner can say, oh, well, this paper says that this particular cell line's a bit weird and it doesn't behave how human cells would behave. So as long as there's nothing out there like that would cast doubts on it, then yeah. Anything else?
Okay. Sarah, we'll send the students any documents you may have to
Excellent.
presentations. And I don't know if it's okay with you that if they have any doubts, they can maybe send you an email.
Yeah, please share
Okay.
my email address. It's fine.
Okay, that's right. Luz, you want to close? You're mute, Luz.
Yeah, here I am. Sorry, my computer is not working with me today. Well, thank you so much, Sarah, for joining us today. It was very, it was great to hear your presentation. So before you go, I wanted to invite you all to our next Nucleate Path webinar. Spellmeyer, here it is. The name of the event is Science is a Good Place for Ugly Little Ducks, and he will explain how NGS revolutionized genomics and how multidisciplinary thinking continues to shape the future of biotech. This is August 7th at 3 p.m. Argentina. So for anyone who is interested, here you have the QR code to sign up for this talk. And here are some useful links. Here we have our social media, our WhatsApp community, Instagram, LinkedIn, Linktree with all of our news. So again, thank you very much for being here today, and we will see you on the Nucleate webinar with Pascal. Thank you so much for joining us.
Thank you, Sarah.